Okay, welcome. This is the second lecture on high-resolution fMRI. It's entitled Functional Contrast and Processing Strategies at High Fields and High Resolutions. And this is a title that Peter gave me, so I will try my best to fit this into one lecture. It could not only be 2 lectures of functional contrast and processing strategies, but I think entire 2 lecture series. So I want to give you a bit of background material and mention it beforehand. So if anything that I tell you today is of interest to you, then you can check out, for example, this review article, which is talking about contrast in high-resolution fMRI, specifically also bold and non-bold contrasts. The processing part, the second part of the presentation, will focus on kind of the concepts and kind of the rationale of what we need to watch out and what people do in layer-dependent fMRI. If you are more interested in kind of the hands-on aspects of that, and there are online tutorials out there where you can, in your web browser, use your analysis. You can download specific standard data and follow, click along to do high-resolution image processing. As I said, there's a lot of material out there. I think the most comprehensive material out there is this 4 days educational workshop from last fall, which is not only talking about contrasts and analysis and processing, but then also kind of potential applications and the use and biophysical underlying processes that we think about when we do layer-dependent fMRI. If you want to have the recordings, let me know. So why do we care about high resolutions? I talked a bit about that already 2 weeks ago. Just as a reminder, especially in this audience, I think most of us try to understand how the brain works. So most of the applications of high-resolution fMRI would be basic cognitive neuroscience. There are many other applications for high-resolution fMRI too. And basic cognitive neuroscience basically bridges a lot of different spatial scales, all the way from the microscopic side, where we have the synapses and the cell bodies, all the way to the macroscopic side, where we have behavior, manifestations of mental disorders, and also the different brain areas. The mesoscopic part here, between the microscopic and the macroscopic part, is kind of underrepresented, and only slowly methodologies are being developed more and more to, like, for example, see hundreds and thousands of neurons at the same time, but also with high-resolution fMRI, not only to see these different brain areas, but also distributions within those brain areas. The keystone and the spatial scale that will bring everything together and then will allow us to build on top of that in a more stable fashion is this mesoscopic regime of these few hundred micrometers. And at this spatial scales, we have the basic neural building blocks, like the layers and the columns. But it's very small from an fMRI perspective, or it's very, very large from these optical perspectives. The reason why we think it's important to get to these few hundred micrometers is because there are different circuits across these different layers, as I kind of alluded to already 2 weeks ago, specifically in kind of hierarchically organized brain systems. So, for example, in the visual system, where we know that the input comes in through the eyes, goes through the thalamus to early visual cortex, and then is processed step by step to higher and higher feature extractions. In those kind of hierarchical brain systems, we know that there's this canonical microcircuit, namely that most of the feed-forward input that comes from this bottom-up connection terminates in the middle cortical layers, whereas feedback input from other areas that are higher in the hierarchy comes in mostly into superficial and/or deeper layers. So by knowing this kind of connectivity of how the neurons are wired, and the circuitry allows us to not only tell if there's this brain area is involved in a certain task, but also then if we know which layer this activity occurs, allows us to tell something about if it's feed-forward or feedback driven, where this activity is coming from. And this is kind of a simplified model that works in some strictly hierarchical organized brain areas. There are also other canonical microcircuits with respect to layers, for example, in the frontal cortex, where it's not so strictly hierarchical. And there are other simplification models out there, for example, this input-output model. And 2 weeks ago, we talked about kind of input and output in the primary motor cortex, but then there are also other things like the dorsolateral prefrontal cortex that I will talk about as well. So after this introduction and reminder why we care about high resolution, let's jump into the 2 aspects, the functional contrast and then later the processing. Let's start with the functional contrast. And I showed you this little cartoon already before, and that kind of reminds us that we are not really measuring neural activity directly. We care about neural activity, but what we are measuring is kind of a response of that. When the neurons fire, they trigger the vessels to open up, to deliver more oxygen and glucose, and then we have these blood volume changes of these large arterial vessels, and therefore by later also these oxygenation changes in the veins that kind of pass through here. So we are really relying on a lot of kind of coupling mechanisms. BOLD is a very complicated signal, and which I want to summarize with this little flow chart over here. So when we have a tissue activity, it triggers a lot of different other processes. For example, then we have arterial dilation, we have oxygen consumption, we have all kind of fluid dynamics until we have actually oxygen oversupply, as we learned from Peter. And this oxygen oversupply, due to the different dia and paramagnetic properties of hemoglobin, results in a susceptibility change. This results in dephasing. This results in a T2 star change. T2 star is this tissue parameter that Vinay talked about. And ultimately, this then translates to our MRI signal change. So the thing that we measure is very far away from the thing that we care about. And there are all kinds of things that can happen along this cascade, right? So an example is that when a participant drank coffee, right? When they drink coffee, the baseline blood flow is different. Your venous baseline oxygenation is different. And this means that at the end, your susceptibility change functional response is like hugely amplified. It can be double as big when you drank a lot of coffee. And then also your BOLD signal change at the end will be huge. And this has nothing to do necessarily with the neural activity. It's just a different fluid dynamic. Or for example, when there are people who are very kind of sporty and fit, they also have a lower venous baseline oxygenation, which changes the BOLD signal, completely independent of the neural activity, which is a problem, right? We want to have something that we can quantify. So ideally, we want something in the first row over here. If we cannot get neural activity directly, at least get some, let us get arterial vasodilation or directly oxygen consumption. Being a physicist myself, I think ultimately the ultimate unit of measuring activity always comes down to energy. So oxygen consumption would be close to that. The second big issue that we have with BOLD is something that I alluded to already 2 weeks ago, namely it's the localization specificity. So I showed you this animation where when you increase the resolution further and further and further, you still end up with these kind of fuzzy blobs. And now you have these blobs at high resolution, which doesn't buy you that much. And we know that this is coming from these cascades of different coupling mechanisms. That basically the signal at the end comes from susceptibility changes at the large draining veins. And the oxygenation changes have actually moved from the location of the neural activity further downstream and are now in a different location. So rather we want something that's much closer to the neural activity, something early on here, for example, like blood perfusion or vasodilation or the oxygen consumption. So we have different sequences out there that are commonly used, also commonly at NIH, and I would like to go through them. One of the methods is looking at VASO, which is trying to look at blood volume imaging. VASO is a non-invasive method. It's aiming to look at blood volume changes, and it's actually a quite simple method in the sense that it assumes that each voxel consists of blood volume and the rest, neurons and gray matter tissue, which is exemplified here as two magnetization vectors. We have the blue one that represents the tissue, and we have the red one that represents the blood. And then in this sequence, in VASO, we start every TR with a global 180-degree inversion pulse that flips around this magnetization. And then the different magnetization vectors are relaxing with T1 relaxation. There's one exact time point right about now when the red magnetization vectors was just going through 0. So during this time, we don't have any blood magnetization available. And during that time, we play out the excitation pulse and acquire our signal. So right during this time, we only see tissue. We see nothing else. So if we have a voxel like this that consists of blood and tissue and all kind of stuff, we actually end up to only get signal from everything but the blood. Black means no signal. Now, what this allows us to do is then do functional activation studies where we know that the vessels open up. And this means that we have less signal in our voxel. We have more black blood in our voxel. And this means that when there's a blood volume increase, we have a corresponding MRI signal decrease. So VASO is a negative contrast, and it doesn't require contrast agents. Other methods that are popular specifically also with high resolution imaging and also to some degree here at NIH, is ASL. ASL stands for arterial spin labeling, and it's designed to measure cerebral blood flow, CBF. And the units of CBF are actually not that different to blood volume. Blood volume and blood flow are highly connected with each other. And the difference between blood flow and volume is that the blood flow measures the amount of blood volume that's delivered per unit of time. So it's like a temporal derivative of the volume. And how this usually works, for example, at some sequences that we've been doing here, is that we try to capture this inflow of blood that perfuses the brain. And for example, we are interested in imaging slices like that. So what we can do is we play out this kind of spin tagging, this 180-degree flip below the slice that we are interested in. And now all the blood water that is located down there and is ultimately feeding the slices that we care about is now kind of labeled. It has a different magnetization. It's inverted. And basically, we invert everything down there. We wait a while until the blood is coming into our imaging slides. We actually wait a bit longer until the blood water magnetization is going through the permeable capillary walls into the extravascular space and deposits signal over there. So at the end, we are looking at a signal that's proportional to the delivery of blood. So we do this spin tagging down there, and then we compare this with an image where we didn't do the spin tagging, we subtract it, and at the end we measure blood volume. And to give an idea how images look like, I'm showing you this example here, where we're looking at 0.9 millimeter resolution, which allows us to look at cortical depth. And I'm flipping back and forth between bold and blood flow. And you can see that it's different. Blood flow has less activated voxels, which could mean two things. It could mean that, well, it's more locally specific. It doesn't have all these false positives, sort of the large draining veins. But also it could mean, well, it's less sensitive, so it has more false negatives. And obviously, I think at the end, the truth is somewhere in between. If you want to get anywhere close, for example, to what we are used to with grain echo bold or vaso, and if you're willing to scan a lot, like many hours, this is double-digit number of hours, then you can also get consistent signal without this danger of the false negatives. And you can see how blood flow in blue kind of avoids these large draining veins at the cortical surface at the border to CSF that is so sensitive in gradient echo bold. Next and last, the other non-bold contrast that's kind of popular and being investigated for layer-dependent fMRI and high-resolution fMRI is calibrated bold, a measure that we are using to estimate something like CMRO2, the oxygen consumption. CMRO2 stands for cerebral metabolic rate of oxygen. And I wish I would have my notes. We know that bold is complicated from this flow chart that we had. And basically, we can parameterize bold as a function of oxygen consumption and the fluid dynamics, the vascular physiology, which would be blood volume or blood flow, which have a tight coupling together. And using these parameterizations that rely on assumptions, physiological assumptions, allows us to estimate CMRO2 by using a series of experiments, namely, for example, a calibration experiment and then the actual neuroscience experiment. So during the calibration experiment, we are, for example, giving the participants more oxygenation, more like a different gas mixture to breathe, for example, air with enriched CO2, which allows which tells the vessel to open up directly. So we have a vasodilation. However, we don't have an oxygen consumption because the brain is not doing anything. We're basically shortcutting, wiring the vascular response function by telling the brain that it wouldn't have enough oxygen, and CO2, hypercapnia, is a very strong vasodilator. And you can see that the bold is extremely high. Blood flow, blood volume is kind of a moderate response to that. Now, then in the second experiment, we are interested in what the brain does during the neuroscience task. For example, during tapping here, we would have a vasodilation as a function of the increased neural activity, and it triggers the vessels to open up. And you can already see that there's something different here among those experiments, right? We have a stronger bold response compared to vascular physiology for the calibration experiment. But for the tapping, we don't have that, because here the additional CMR02 actually reduces the oxyhemoglobin against in a sense that now the bold and the CBF, CBV are more comparable. And you can already see that basically with a set of equations, you can factor out the oxygen consumption. You calibrate your equation, and then you can tease out oxygen consumption by assuming a number of physiological laws. How this looks in practice is then here for a finger tapping experiment where we are interested in the tapping right over there, where for blood volume, we see these kind of stripes over there, similarly for bold, and then for hypercapnia, we see everything being activated because globally in the entire brain, all the vessels open up. So we have different layer response functions, and then we put them into the model, and then we get these different peaks that should be independent of any vascular physiology. While this approach has been used for maybe a handful of layer fMRI studies out there already, it's still being kind of investigated how valid these underlying assumptions are, how if this model that has originally been developed for low resolutions really applies also at high resolutions. To compare these different methods, I tried to look at them with respect to quality metrics that I find useful for any kind of high resolution fMRI sequence. Namely, first, we need sensitivity. We need to see that there's signal changing. We need a localization specificity. It needs to be non-invasive. Like if you need to give a person like an injection, or you need to ask them to breathe weird air mixtures that makes them anxious, it's not the best thing. How available the sequence is, sampling efficiency, and quantifiability. And for gradient echo bold, indicated here as these color changes of oxy and deoxygenated veins as blue and purple, we have huge sensitivity. The localization specificity is pretty poor, so is the quantifiability. It's not invasive, every scanner has it, and it's incredibly efficient. Next, let's look at ASL, for example, which is not as sensitive. I think it's the most locally specific among all of them, because it's not only looking at arterial dilation, but it's literally looking at the capillaries that are really close to the neural activity. It's kind of available among most scanners, but it's still not as easy to use. You need to understand the sequence a bit and then analyze it, and more cumbersome compared to gradient echo bold. Next, let's look at calibrated bold, which is really shining in with respect to neural quantifiability. The rest of them is kind of not ideal. With layer specificity, we don't really know. It's kind of as good as the models are. And last, BASO. It's not as sensitive as others. It's not as quantifiable as others. It's not as available as others. It's kind of a compromise among many. It's definitely more sensitive than ASL or calibrated bold. So having talked about all these non-bold contrasts, let's summarize that they are all noisier than gradient echo bold. They are all kind of more locally specific, and they're all kind of more quantitative. You can better compare different brain areas or different people. But there are more sequences, especially in the early days of 7 Tesla and high-resolution imaging. There was a lot of talk about spin echo. And there are many, many different flavors of this. If you do it with diffusion weighting, if you use it with an EPI that has T2 prime and non-spin echo contaminations in there. I don't want you to go through all these different sequences, because spin echo has lost a lot of its popularity, I think, in the last maybe decade or so. There's still people using it and playing around with it and trying to improve it further with some success also. But ultimately, the take-home message I want you to remember is that there's this trade-off between localization specificity and sensitivity. And if you want to be really, really, really sensitive, you're usually often picking sequences that are sensitive and then giving a sensitivity that you might not necessarily be interested in. For example, the large straining veins. If you get rid of the large straining veins, then you're not as sensitive as more, but you're more locally specific. So it's kind of a trade-off space. And there are additional sequences out there as well. And while the field has been fighting a lot in the last or early decades of high-resolution fMRI, which sequence is the best, I think these discussions have kind of calmed down in the recent years, because people realize that there is no best sequence for high-resolution fMRI. it's kind of like this quartet card game, and I'm not sure how popular it is in US, where you basically have different metrics, and some of them methods shine with some of them, and others shines with others. So asking which sequence is the best is kind of like asking which vehicle is the best means of transport, right? It depends where you want to go. And very often, especially for long distance travel, you use a combination of different methods, which I think is now has evolved into the standard. If you look at all the high resolution papers out there, there are about 250, then about a quarter uses gradient echo bold and kind of ignores the quantifiability and localization specificity issues. About a quarter of them uses gradient echo bold and accounts for them to some degree. And about half of the papers out there use a combination of methods where they benefit them both from gradient echo bold as well as VASO or other non-bold methods. There are even more exotic methods out there. I found it quite hard to have an overarching complete list of sequences that people use for high resolution fMRI. For example, there's functional diffusion actually quite early with exquisite results. And then there's kind of spin locking sequences to also look at CSF contributions to layer-dependent fMRI. And then there are also these direct neural imaging methods that I'm not sure if Tyler will talk about it in a few weeks, which also look at very high localization specificity across layers. Let's go to the second part of the presentation, the processing strategies. And with respect to processing strategies, I would like to start with kind of pre-processing, how you deal your raw data, and then later post-processing, how do you extract useful neuroscience informing information out of your data. And with respect to the early processing, how you deal with the raw data, layer fMRI and high resolution fMRI is really challenged a lot. And with respect to that, I want to pitch this software suite that actually comes originally from NIH and is now mostly maintained and professionalized by Fru Gülban in Maastricht as part of Brain Innovations, which tries to solve a lot of issues that we had in high resolution fMRI in the early days, which I would like to list now. So for example, in high resolution fMRI, as I said in the last lecture 2 weeks ago, we are having a kind of compromise space where you can either have a lot of coverage, high resolution, or fast sampling. So usually you want to optimize your acquisition protocol for your specific question for your brain area. So usually in high resolution fMRI, it's just wasteful to acquire the entire brain. You don't want that if you have a question about a specific brain area. So people have only these labs. These labs, however, have issues with respect to analysis because they don't fulfill common topology requirements of a brain, namely having meshes that are closed without holes. So specifically for these labs, if you try to do layerification, we have these kind of unconnected dots. On top of that, we have the issue of the beer-wine dilemma, how I call it. Namely that high-resolution fMRI is really optimized for each protocol, and it's not as kind of established and fine-tuned as conventional whole brain fMRI. So in whole brain fMRI, that's around for decades, there are standardized pipelines where you can pump through your 1,000 participants through them, and for most of them, it will just work, like done by multinational brands of beer corporation. In layer-dependent fMRI, we don't have such pipelines. I feel more like wine, where every study has their own little tweaks. There are no huge standards of international brands of wine corporation because each country, each weather, each year needs its own little tweaking to get most out of it. And with layer-dependent fMRI, I think we need to have a lot of user-dependent flexibility to optimize and fine-tune your analysis. Other things, as I said already, we need to deal with these non-bold contrasts or other forms of imaging contrast that then ultimately needs to be analyzed respectively. And also in layer-dependent fMRI, very often we have different artifacts and we have just a lot of noise, which we need to deal with. So we need noise cleaning methods or these artifact detection tools, which are all things that LANI provides us to some degree. One of the most often used feature in LANI, this layer fMRI program, is layerification. And by layerification, I mean basically the mathematical problem of having tissue borders of gray matter to white matter and gray matter to CSF, and then dividing the cortex into different chunks of different cortical depths. And this is a very high resolution ex vivo scan with cortex where you can see this dark line. This would be a layer 4, striagenari. And you can see already that it's kind of biased among curvature, that at the top of a gyrus, it's kind of very, very close to CSF. At the bottom of a sulcus, it's very, very close to white matter. So if you just take the distance of gray matter to white matter and divide it into 2 parts, it's not really capturing these kind of orientation and curvature biases. And for example, here we are on too far on the left, here we are too far on the right. So we need these equivolume layerification algorithms, which seem to describe very well how the biology kind of divides the different layers, namely basically having different chunks of tissue and then having a layer referring to a cortical depth by means of volume units. And we want to have this specifically also flexible enough for these kind of, without really for requiring common topology requirements of mesh-based layerification, namely having closed surfaces without holes. And this is an example of that I showed you 2 weeks ago already, very high resolution data where this was a multi-session experiment, and then you couldn't really align the different slabs perfectly. So we never had perfect meshes. And there was a lot of manual editing necessary. And then in the area of interest, without really worrying about anything else, you can go in there, manually correct it, and then run your layerification in there. Other things that are always necessary with respect to analysis in high-resolution fMRI is kind of the columnarification, where you basically look at the brain in kind of patches of topography. Basically, if I'm zooming in, and I showed you this example 2 weeks ago of the primary sensory motor system, you want to have a coordinate system where you're not only looking at cross-cortical depth, but also then in the other 2 spatial dimensions. And how this is done in LANI is you chunk up the brain into smaller and smaller and smaller pieces, look at the neighbors, and then flat it out. And ultimately can look at across depth of a sulcus, lateral columnar distances, as well as kind of across cortical depth to look at these mirrored finger representations that we talked about. Other things that are quite common in layer-dependent fMRI, specifically in studies that are only using gradient echo bold, is then to account for the veins. And we know that veins always come from the surface in the tissue, and they're drained in the other direction. So the oxyhemoglobin that's changing its susceptibility is generated at the layer of activity and then drained towards the surface. So we always know that the superficial layers are not only representing the neural activity of the superficial layers, but basically a weighted sum of the neural activity within that layer and everything in beneath. Additionally, we also have different vascular gains. There's just more vessels in the superficial layers than the deeper ones. And you can model those, parameterize those, and then account for it. For example, just getting rid of a linear trend across cortical depth, which gets rid of a lot of these false, like strong activities in the surface only, makes it more flat. Then you can also try to scale it with the gain, and then also try to deconvolve it spatially. And for each step, you kind of see that you are removing a lot of signal that you originally had in gradient echo bold, which makes this, the activation maps a bit noisier. And this is called deveining to get rid of the vein effects. Other things that you can do in a layer-dependent fMRI, specifically for debugging, is to smooth your data, but you don't want to smooth them isotropic in all directions, because then you would lose all the high spatial resolution that you were after. But instead only smooth within the layers. So smooth all voxels that are in the same cortical depth without signal leakage across cortical depth. That can turn kind of these very ugly single run speckle noise into nice activities that follow the cortical ribbon. Let's jump to post-processing. How do people make sense of layer-dependent fMRI data in order to tease out information that they cannot see with conventional imaging? And I think the vast majority of studies out there, it's just looking at one brain area, they're zooming into that cortical area, and then use an approach that you use with conventional kind of fMRI location mapping, basically looking at task subtractions and try to quantify at which layer these task subtractions are bigger or smaller. And I would like to explain this on the example of a study that Emily Finn did here at NIH, where she was looking at the dorsolateral prefrontal cortex. And from the monkey literature, she knew that there are different types of cells in there. And there are these cue cells, there are delay cells, and then there are these response cells that ultimately trigger the motor systems. And we knew that all these different cells are located across different cortical layers. So she tried to modulate the activity of those different cells by showing participants these strings of letters and then asking participants to either memorize the order of their letters or mentally in their mind sort these letters according to an alphabetical order, which is quite hard to do if you only saw it very, very briefly. And then a short while, and this alphabetization is believed to mostly engage the upper layers, cue and delay cells. Later in the trials, then participants had to do a response decision based on the location of a given letter with or without the alphabetization, which is then believed to mostly engage the response cells that trigger the motor systems to press a button. So in conventional layer-dependent fMRI, what you do is just contrast the different task conditions and then look at, for example, the alphabetization versus remembering or the responding time series, where you can then see and map out that the different response conditions are differently distributed across cortical depth. And if you're lucky, then also there were distinctions across time points when these different parts of the trials were referring to the upper and deeper circuits. Other things that are being kind of explored in high-resolution fMRI, how to make sense of high-resolution layer-dependent fMRI, is also referring to resting state connectivity, which is coming back to the hierarchical organization of some brain systems. So I told you already that especially in the early visual areas, we have a very strict hierarchical processing, meaning that an area like V1 receives feed-forward input from thalamus, while also V1 receives feedback input from other areas that are higher in the hierarchy, for example, V5. And in this very simple test case, proof of principle studies, we had localizers of the LGN, actually Pulvinar a bit too, then V1 and V5 based on structural and functional localizers. And then when you look at your layer-dependent functional connectivity strength in V1, you can see that you get different profiles depending on which seat region you're grabbing. So here, looking at layer-dependent responses across depth, the surface should be on the left, close to white matter should be on the right, which is missing, I apologize. You see that for a seat region in thalamus, most of the fluctuations that happening in the thalamus seem to be correlated with the middle deeper layers. The superficial layers in V1, they don't really care what the thalamus is fluctuating about. This is different in the same target region, in the same V1, when we pick a seat region in V5MT, because then we still have these kind of shared covariants, but also we have this additional source of fluctuations that's coming from feedback from V5 to V1, which might also be visible as these kind of off-diagonal elements in a layer-by-layer correlation matrix. Pushing it even further, trying to explore what kind of analysis strategies might make sense with respect to layer-dependent fMRI. I also want to show you this kind of exploratory analysis where we are trying to understand the usage and the usefulness of movie watching paradigms in layer-dependent fMRI. So here we are using the same movies that are used from the HCP, the 70 HCP dataset, where they have many, many participants. And then if you look at those participants, you can see that there are different subgroups and there are individual differences. Now, using these same movies and having the data of low resolution across the whole brain also allows us then to do kind of functional connectivity mapping across the conventional networks here, for example, the default mode network, but also more clear actually were the results in the task positive network, this frontoparietal network, where in this network, using the HCP data, we saw that there are kind of 3 different populations, or we forced it to be 3 different populations. And then you can see that the time courses are actually quite consistent for watching these 6 different movie clips. And then there are time frames that are more stable across people, and there are time frames that are less stable across people. So now, for example, here, these nature scenes were more stable, these kissing scenes were less stable, people reacted differently in that network. And then looking at those different time courses with high-resolution fMRI, we can do inter-subject variability studies and see that these differences are almost exclusively coming from the superficial layers, which, on some sense, I think makes sense if you think about that all the people see the same movies. So the retina has the same input, that the low-level features are identical, and then the signals are processed further and further and further and further until you get to higher and higher cognitive areas. But at some points, these inter-individual differences come in. Everybody perceives the movie differently, whereas kind of the input is the same. So it makes sense that these inter-individual differences are mostly coming from the superficial layers. As soon as you kind of explore layer fMRI to further and further analyses outside of these strict validation experiments where we kind of know what to expect from the animal literature already, but trying to also move this to more cognitive areas, we have the issue that we kind of need to relate the layer-dependent, depth-dependent signals with respect to layers, which are different across different brain areas. So you know that most of the people refer to 6 cortical layers, the 6 Brodmann layers. There are other definitions though, and which are kind of almost in all different brain areas somewhat represented. However, across different brain areas, they have different thicknesses, which is originally the reason why we have different brain areas. Like Brodmann defined the different brain areas based on their cytoarchitectonic laminar differences. This tells us, however, that if we want to compare, like, for example, activity in layer Brodmann area 32 with Brodmann area 24, we might have a different kind of layer profile. And simply because the cytoarchitecture is different. For example, here in those areas, layer 4 is just much, much thinner. And in order to calibrate and make sense, how to make sense of these functional data with respect to cortical depth, by thinking about layers, it's very helpful to have these ex vivo normalizations. Basically, there are a few data sets out there that do histology, slice up the brain and look at it with histology to know where the layers are. And at the same time, in the same brain, before they sliced up the brain, we're acquiring very high resolution ex vivo structural MRI. From the structural MRI, you get these kind of landmarks of dips and peaks that you can directly in the same space refer to the cytoarchitecture. And then as soon as we have this translation and relationship, we can then also look at these dips and peaks in humans, so we understand where our functional responses are coming from. More high-level analyses that people are exploring are things like representational similarity analysis, just at the beginning in the first steps to understand what the issues is and to quantify the capabilities and challenges. And this is, again, using the same movies that I showed you earlier. Now, this is a data set where the same participants have been scanned 50 times. And then for each movie frames, you can extract a whole brain activation map. And then you look at the similarity of the spatial pattern of the whole brain activation for a given movie frame with respect to all the other frames across the movies. And that you get these kind of representational similarity analyses. And you can see it's kind of messy, but there is some structure there, right? There are just some clips that are more dissimilar than others. And it might be better visible also when you just average the similarity scores within the different movie clips. But you can see that there are just some movie clips that are more similar to each other or more similar represented to each other in the respective brain area as opposed to others. And this is also something that you can then compare with behaviors. For example, you can just ask people, do you think that those 2 movie clips are more similar or those 2 movie clips are more similar? And then you rank it and you get a similar matrix out of it, where again, you can see that some movies that are represented in the brain to be more similar are also in the behavior claimed to be more similar. This, however, is kind of very specific to the brain area you're looking at. Here, for example, for these visual areas like LOC that are kind of feed-forward driven, where we have the highest similarity in the middle layers, and they match the behavior quite nicely. But then there are also other areas, that are not really matching this behavior, where we don't see this kind of bright, dark pattern. And those are areas that seem to be more superficial driven, where most of the similarity occurs in the superficial layers. All right, getting to the end. Other things that people have done with high resolution fMRI are modeling work of computational cognitive models, such as population receptive field modeling. And Maybe you have heard about this already. You will definitely hear about this in the summer course already, where for these receptive field models, you basically can see for any given voxel a certain bold signal change, and then you build a model, a Gaussian model, where you say that for this voxel with a certain spread, how would the signal given that stimulus look like? And then you basically can do a forward model that for this voxel, we have a given response across time. And for this voxel, it, for example, would look like that, which kind of looks matching sometimes, but then other times not so much. And you can move around and see which location and receptive field site best fits the time course that we're actually measuring. And in this case, a voxel like here would best predict these kind of fluctuations across time. So for each voxel, you have two fit parameters, that the location, x, y, and then also the receptive field size. And from the receptive field, we know from the animal literature that we should expect a certain distribution across cortical depth. For example, for the receptive field size in red, we see that the deeper layers should have the largest receptive field size, a minimum in the middle layer, and then at the superficial layer, it increases again a bit. And Alessio Fracasso, for example, has looked at that, as have others, and kind of see similar things in receptive field size across cortical depth as expected from the monkey literature. I think it's monkeys. Is it, Tyler? Yeah, Tyler's nodding. As a very last slide, I think reflecting on high resolution contrast and high resolution processing, there are many, many things that you can do. There are many things you can do wrong. But if you understand the kind of the disadvantages, the biases in your analysis or in your contrast and in imaging processing, you can get very, very stable results out of it. Here, for example, looking at this kind of bimodal hump in the primary motor cortex, you can get this with blood flow, with bold. You can get this with connectivity. You can get this with other contrasts that I didn't even touch on, arterial blood volume imaging, with phase regression. You can do this with vertex-based layerification or voxel-based layerification. If you know what you're doing and you account for veins and the things that you know are your compromises, then I think you can get very, very stable results out of it. So there's no kind of best method out there as long as you know what you're doing. And with this, I thank all my colleagues and I thank you for your attention.